VISTA is software tools for comparative genomics. You can see the website there. We're going to use this pre-computed alignment to compare the human genome and the chimp genome. Pull up that browser. It defaults to the mouse genome. We want to compare human and the chimp genome. That's what you're seeing in front of you right now. We only want one track to display at a time. That's the comparison between those two genomes. We want to look for the HAR1 gene, and we got HAR1A and B. We want to select either one of those, and what we'll do is we'll expand by zooming out first and finding the region where HAR1A and HAR1B are. And so go ahead and zoom in on that region. And now what we want to do is use this browse alignment tool. That red box shows the region that's indicated at the bottom, a letter by letter comparison between the human and chimp genome. That red box is a SNP. We'll talk about that a little bit later on. We want to look at the alignment, and that puts out a direct comparison between the top line, which is the human genome, and the connected line below it are the corresponding letters in the chimp genome. So human on the top, chimp on the bottom. Notice there's a almost perfect match, and here's a region where there is a mismatch, a G instead, and is in the human genome, a C is in the chimp genome. So let's go ahead and copy this text. I'm going to copy this text and dump it into a text editor. On my Mac, I just used the program TextEdit, and dump all of this text, which represents the complete HAR1 gene, into that text file. So I create a new document, paste it in, and there it is now, just verbatim in a text file. I use this because it's a good searching tool, and what I want to do is search and find just that 118 base pair residue that was indicated in the article on scanning the genome. So let's see if we can't find by first looking at the portion of that top sequence. So starting with TGAAA. And so let's just go ahead and use the find button, the find function in TextEdit, and just go ahead and start typing in TGAAA. And then eventually we see that when we get to the second G there, we've got only one piece of that entire genome sequence that has that letter code. So there it is. That's going to be the start of the 118 base pair sequence. That is the part of the HAR1 that is indicated as undergoing an accelerated amount of change. There you see a mismatch, a C with a T that's indicated in that figure. So if 61,733,505 is that A, we can go back and find that T and actually look at that as being a few bases backwards. We can count them, and that's going to be 617,33,494. So if we just go ahead and do that counting, you can convince yourself that that's the case. Now we're going to add 118 base pairs to see how far that goes. 61,733,612 should get us to the final portion of that 118 base pair sequence. Let's go ahead and type those numbers in back to the beginning of the VISTA tools, and we're going to go ahead and use that VISTA browser, the Java 2 enabled function. We want the human comparison to chimp, so we don't want the mouse. Go ahead and pull that chimp genome up, and we'll go ahead and use the defaults with one track shown at a time. And what we want to do now is take a look at what that region is that's displayed based on the coordinates that we typed in. And as I scroll across, you can see it ends on 612. So we've got the right sequence. We can do that direct comparison. That's at the bottom. We can compare it to that figure. I'm looking at the very end of that sequence, the last few letters, the last 6 letters or 7 letters. And you can see that G mismatch. In the chimp, it's an A, and